Well, hello again, human biology class, coming to you online again. This is for Friday's lecture. I'm not sure if I'm going to get through everything that I wanted to on Wednesday, but it was a good break. So what we're going to do in this particular video is we're going to start talking about cell theory. So we're going up organizationally, we're going up the ladder a little bit. We started talking about atoms and then molecules, and then we went to organic molecules and talked about those, and now we're going up organization a little bit higher to actual cells. So the cells that the organic molecules might come together to form, along with other things. So cell theory. If you look at your notes, it says cell theory. All of these things are composed of cells, and new cells only come from pre-existing cells. Well, that may seem fairly obvious, but if you think about what some really smart thinkers thought about such matters before they had the advantages of microscopes and microscopy. For instance, Aristotle, he thought that it was a readily observable truth or fact that aphids arise from dew, which falls on plants, and fleas come from putrid matter, he thought. And he thought mice come from dirty hay. They somehow just are produced by dirty hay, and that flies come from rotting meat. He really didn't have a good understanding of the microscopic egg that a fly might lay, or how mouse reproduction occurs when he's not looking. Just didn't have benefit of that observation. So it's important to say what our disciplines consider to be central logic, things around which the discipline organizes itself. And cell theory is one of those. Atomic theory we've already talked about. So cell theory is another. We'll get a little bit into gene theory here also. That's really important for biology. I want to introduce you to a gentleman that I had known. His name is Waldo McBurney. If I can get to it here, I got to move me out of the way a little bit here, and then we'll go here. And there's Waldo. Waldo McBurney is an interesting gentleman. He actually passed away a few years back at the age of 106. You see these medals on the wall back here? Those are from the Senior Olympics. The last one he participated in, he was in his early 90s, and he participated in at least two events, one of which was a jumping event. I can't imagine that. And the other was like a 200-yard dash or 400, I don't know what it was. At any rate, he lapped his next closest competitor. I talked to him about that, and he said he wasn't going to compete in any more Senior Olympics because he was tired of competing against all those 90-year-old kids. And he's got a unique problem, and that is that when he got married shortly after World War I, he lived with his wife for over 30 years, and she died. No, he was born shortly after World War I. And anyway, she died in like 1960, I think it was. And so he had a tombstone made, and with her name and birth and death date and his name and birth date. And he went on with his life. Well, a few years later, he met another woman, and he's now been married to her for over 30 years. And so everybody said, well, what are you going to do? It's like, where are you going to be buried, next to the woman you're with or the one that was your first wife? And because his tombstone, he has another problem, because the tombstone that they originally made, his wife died in 1960-something, so they put 19, and then they were going to fill in the eventual death date for Waldo, since they would never dream he'd live to 2000. Well, here it's in the early 2000s, and he's still alive, so here's a tombstone with his name on it, and 19, and with a woman that he hasn't been married to for over 30 years. And I kind of thought, it's really not his problem. It's somebody else's problem, because once he dies, that's somebody else's decision. The whole point of this, though, is why do people like Waldo McBurney live so long? He lives well. I mean, I knew him at this stage, and he ran a bookkeeping business, he ran a seed cleaning business, and he ran a honey business. 3 different businesses. He was. I think in this picture, he's over 100 here in this picture. And if you saw him, and he still got his teeth and everything. If you saw him, you'd think, middle 70s. But no, he did quite well. He did eventually die at 106, as I say. So what is there about him that allows him to live so long, and other people that, they're old in their 50s and don't live past their 60s? And so he's participating in a study that's looking into that type of thing. Sorry about that brief pause there. I wanted to make sure to get the notes visible here for you to see. And so here we have those notes. So we're at cell theory. And really we're talking about two types of cells. And those are prokaryotic and eukaryotic. And mostly we're talking about eukaryotic cells, cells with a nucleus. Prokaryotic cells are also important, like viruses and bacteria, because they affect us, so we won't ignore them, but we're gonna concentrate mostly on eukaryotic cells. And the first thing we're going to look at is the cell membrane. Okay, so if we, that's not the one I wanted. So if we go to the cell membrane, well, first the cell. Here's your rendition of the cell from your textbook, and the membrane, of course, bounds or encloses the cell. And if we look specifically at the cell membrane, that's what we have here. There are some structures that are really important to point out. When we talked about lipids, we talked about a phospholipid. That was a lipid that was a little bit different. It wasn't a triglyceride. It only had 2 fatty acid tails. And the third fatty acid was really this phosphate group that's displayed here as a round knob on the top of that particular molecule. So this is not a glycerol head right here. They might have a better picture yet. Let's see here. A little bit. Oops. Just did the wrong thing there. Bear with me. Right here. This one is a picture also from your textbook. And here are the phospholipids. Again, it's confusing. I wish they hadn't done this. But you'll notice each of these molecules has 2 tails. Those are each a fatty acid tail. And this is not a glycerol head. This is the third fatty acid tail having been replaced by a phosphate group. So the bottom line is we call this a phospholipid. And because there's 2 layers of them, we call this a phospholipid bilayer. And that constitutes most of the cell membrane. That along with these big orange things here. And these are big protein molecules that are embedded in that phospholipid layer. And what they really are, this shows it well, is a conduit or a passageway for molecules that these protein molecules will identify and allow in. Now here, they're showing water molecules, and we're going to get that in just a second, but really any molecules. For instance, a really good example would be a glucose molecule, which is bigger than water. And it's going to have to be recognized by one of these protein carrier molecules and carried in or transported into the inside of the cell before it can be used to produce energy. Okay, so the cell membrane then allows molecules in and out selectively, just certain ones in and out. You don't want everything being able to get into the cell membrane or into the cell or out of the cell, just certain things. And there's 2 layers, as I mentioned, of phospholipids. And really the cell membrane, a good way to think of it is like a revolving door. And you know this revolving door, it's basically only going to let certain size things in and out, and it's oftentimes size that makes a difference, and other times it's a thing that has to be recognized by its shape. For instance, this would only let things in that could fit through there because of size or because of shape. So this is a good way to think of the protein carrier molecules and openings that are in the cell membrane. This whole wall would be the cell membrane, and these revolving doors would only let certain things in and out. Good way to think of the cell membrane. So this cell membrane has a hydrophobic and a hydrophilic end. So if we go back to that particular picture from your textbook, so the hydrophobic and hydrophilic end, maybe I'll go with, I think the other picture was a little bit better. We'll see. Oops, I didn't get it fired up here. Okay. So there are hydrophilic is a water-loving side, and that would be the phosphate side, and hydrophobic would be the water-hating side or water-repelling side, and that would be the fatty acid tail side. So it's definitely a barrier that allows for things to be kept in the cell and to only allow certain things to move in and out of the cell. So that's an important aspect of that cell membrane. And I already mentioned the protein carrier molecules, but I want to go back to then, so then I mentioned here that the membrane also has these aquaporins and gap junctions. And if we look here, this one here, an aquaporin, if you look at its name, hopefully it makes some sense. Aqua, having to do with water. Porin, it's like a pore or a hole. And that's what we have here. And they're in the cell membrane to allow water to move fairly freely in and out. Now, if you remember, water is a small molecule. And so not much else can get in or out through that opening in the cell membrane called an aquaporin other than water. There's another similar structure. It's called a gap junction, and this is what it looks like when it's closed. This is what it looks like when it's opened. And this is one cell membrane. Here's another cell membrane. And so it allows for movement of very small items across cell membranes. Okay, so those are structures that exist within the cell membrane, tiny openings. So molecules may move across the membrane in one of three ways. Basically, diffusion is the first one. It's the movement of molecules from high concentration to low concentration. The movement of molecules from where there's a lot to where there's a little. Okay, and if you think about it, that makes sense. If anybody's opened a bottle of perfume and I'll do this in lab, in a room, you know that the perfume doesn't just stay in the bottle. It's concentrated in the bottle, but it moves and eventually is smelled all throughout the room. And that's diffusion going on. I'd also guess that a number of you have heard somebody talk about, don't put your textbook under your pillow at night in hopes that you'll gain the information in that textbook by osmosis, that it will somehow magically move across the pillow and into your brain by osmosis. Drives me nuts. It's a small trip, but it drives me nuts. Osmosis has nothing to do with the movement of information. Osmosis only has to do with the movement of water. Water, not information. And it's that movement of water across a selectively permeable membrane. Selectively permeable means that it's selective. It only allows certain things permeable, to permeate, to move through. It only allows certain things in or out of the cell, or in or out, or through the membrane. So osmosis only allows the movement of water across a selectively permeable membrane. Only water, that's what we're talking about. Well, facilitated diffusion is another one, and that's that. We're going to go back to that picture that I showed you of the cell. We'll go back to this large picture first here. And here's the whole cell membrane. And these are those large protein molecules. You could think of them as a protein carrier molecule. And it's almost like they have an antenna sticking off of them here and here and here. And really that's accurate because if, in some of these proteins, this molecule, it's there to recognize certain other molecules and ignore others. It will only recognize certain molecules. For instance, if the job of this is to recognize glucose, it will only recognize glucose and allow glucose to pass through the membrane, through the protein molecule, and get into the inside of the cell. And so a facilitated diffusion requires these orange protein molecules. No, they're not orange in your cell membrane, they're just orange in the picture here. Allows these protein molecules, depends on these protein molecules to allow recognized molecules into or out of the cell membrane. So it's facilitated. Facilitated means it's helped by. It's helped by these molecules, across the membrane. Facilitated diffusion. You still have movement of those molecules from an area of high concentration to a low concentration, so it's still diffusion, but it's facilitated, or it's helped by these protein molecules. Okay, if we go back to your notes and we look, we're right here. We talked about diffusion, we talked about osmosis, we talked about facilitated diffusion. The last one is active transport. Active transport is the only one of these that actually requires energy. Your body is set up miraculously so that so much of it runs without using energy. It runs downhill, if you will. Things move from high concentration to low concentration. But every now and then, you've got to spend some energy to get stuff where you want it. To get stuff from where there's not that much to where there's already a lot. And that's really what's going on here. And so molecules move from low concentration here now to high concentration, requires energy. Best example I can come up with is when I was a student at Bethel 110 years ago. And back then Bethel owns the apartments across 35E called Fountain Terrace. And my friends and I lived in one apartment, and our RA was a guy named Randy, and he had a crush on a young lady that lived in an apartment across the hallway from us called Kathy. Her name was Kathy. So, you know, we're sitting there in our apartment, and Randy was trying to figure out a way to get Kathy's attention. And, you know, guys will do silly stuff to do that. Crazy stuff. Sometimes not very good stuff. Well, this was I thought it was kind of clever. Randy was trying to figure out how he could get Kathy's attention and let her know that he was thinking about her and cared about her and would like to take her out on a date or something, maybe even. Well, myself and the rest of the guys in our dorm, you know, were geeks, and we didn't have a chance of getting a date, so we thought we might as well help Randy out get a date. And so his idea was, he had access to her apartment, and the approval of her roommates, and so he decided he would fill her bedroom up with newspaper, crumpled up newspaper from floor to ceiling. You tell me why guys think of that stuff. We'll both know then. I don't know. Anyway, so we figured out, well, we'll help him out. And so he comes up with a stack of newspapers. By the way, if you ever want to do this, you want to go with Wall Street Journals. Boy, you get a lot of volume out of those. Star Tribs are okay, but they got a lot of schmutz from ink that can get on stuff. Well, Randy thought of that. So he covered her bed and her furniture and everything with plastic so it wouldn't get newspaper smudging on it. And then we went to it. So we just sat down outside the door or in the bedroom, and we just, you know, grabbed newspaper, crump, crump, choo, boom, boom, boom. You know, just all of us there was probably 4 or 5 of us just crumpling newspaper and throwing them in the pile. It grew and it grew and it grew. And pretty soon it was her bedroom was so full that we had to have the door just open a crack. And still we were throwing the newspaper in through the crack in the door and filled it up even more until we could get no more in, and we closed the door. Then we all ran across the hallway to our room, and they had these little peepholes on the door where we could look out, and we all sat there kind of giggling, you know, waiting for her to come back from wherever she had gone. And she comes back, and she goes into the apartment, and we're waiting, you know, with anticipation, and pretty soon the door opens to her bedroom, and there's this big shriek as all the newspaper came out and fell on top of her, and it just worked flawlessly. And we all just were very proud of ourselves. And it worked really well because Randy ended up marrying Kathy. So you guys can decide, you know, if that was worth it or not. But anyway, that's an example of active transport. After a while, there was a lot of newspaper in Kathy's bedroom, and we kept putting more and more and more in there. And using energy to do so, okay, against a concentration gradient until the door would not even be opened anymore. All right, active transport. There's a video that I'm going to put a link to under this lecture, and at this point, I would like you to go watch it. It's 1 minute and 18 seconds long, and it's on a gentleman by the name of Bear Grylls, which I'm thinking some of you may have seen. And he does something in this video that it's disgusting, but I want you to think about what he's doing, and if in fact, given these aspects of diffusion, facilitated diffusion, active transport, if in fact that makes sense. And then we're going to apply that also to the next topic, which is tonicity, hyper, hypo, and isotonic, and see if this all makes sense. And so go ahead and watch that video, and again, the link will be just underneath this video lecture, and away we go. Okay, so just to finish up here then, tonicity, the distribution of water and salts on either side of the cell membrane. Okay, so if we're looking at an image of this, this isn't the best one. I don't really have anything to represent the salt, or this is from your textbook here. I guess I'll come back to this one. So yeah, if you think about this cell membrane, and the fact that water can move pretty freely across it, but salts cannot. Salts are bigger molecules. When I use that word salts, sometimes I'm talking about sodium chloride, but sometimes I'm talking about lots of other things. Basically, anything that can be dissolved in water can act like a salt. Potassium chloride, magnesium salts of various. There's lots of different things that can be salts. There are 3 types of distribution here. If I start with the last one on my list here, which is bad pedagogically, forgive me for that. Isotonic, well, then the outside of the cell has basically the same number of salt molecules as the inside of the cell. Okay, and that's just kind of steady state. And there's hypertonic, where the outside of the cell has more salt than the inside of the cell, and hypotonic, where the outside of the cell has less salt than the inside of the cell. It's kind of like Goldilocks here in her porridge, you know. One was too cold, one was too hot, one was just right. Well, let's think about this just a little bit more. Hypertonic. The outside of the cell is saltier. Okay, so those are big salt molecules, and they don't move easily, but water can move. So if the outside of the cell has lots of salt, and there's not as much salt on the inside of the cell, well, the salt can't move from the outside of the cell in very easily, but water can go through aquaporins and gap junctions really easy, so water moves out of the cell. What happens? Well, the cell shrivels. And that's what this is trying to show here. Here's a blood cell put in water, okay? And this is trying to show that they should have some representation of salt, this being salty around the cell, so that water moves out of that cell to kind of try to even things out, so there's more water outside, you know, so there's more water here and not as salty, but then the cell shrivels because it doesn't have the water to give it its shape. Well, there's also hypotonic, where the outside of the cell doesn't have much salt at all, maybe even no salt. Well, the inside of a cell has salt in it. So what happens? Well, water moves into the cell to try to dilute all that salt that's in the cell so that it's about the same outside and inside of the cell. Well, what happens though? Well, that cell gets bigger and bigger and bigger until it explodes. It may just pop. It's just showing a pudgy cell. They have a hard time representing that here. This would be a narrow spot in the middle, and this would be more like a balloon. Okay? So that's a hypotonic solution. Now, it's careful to look at the wording here. Hypertonic. Well, what's hypertonic? Well, the outside, the solution that the cell is placed in is hypertonic. The outside of the cell is hypertonic. Hypotonic. What's hypotonic? The outside of the cell is less salty, is hypotonic. So we're talking about the solution that you put cells in, and then isotonic. Okay, so hopefully that makes some sense. And watch that Bear Grylls video again. Refer back to that. It'll come up again, but we'll talk about it in class after the quiz on Monday. Remember, quiz on Monday. No, I think I'm going to move that quiz to Wednesday. Quiz on Wednesday. Cytoplasm. The last two things. I want to talk about these two things. If we go back to that picture from your textbook right here, so here's the cell membrane. We've spent quite a bit of time on that. It's that important, though. All this stuff inside from the cell membrane in is called cytoplasm, basically cell guts, if you will. And probably the thing that stands out the most, what you saw in lab on Tuesday, was the nucleus of your cheek cells. And that's one of the most obvious features. And if we look at the nucleus, a couple things to say about it. Of course, we'll get into more detail on it. But the cell control center is the nucleus. It's porous, and it's itself surrounded by a nuclear membrane. Okay, so we go back to it. You can see those pores here. This is a pore, this is a pore, this is a pore, and it has within itself a structure called the nucleolus. Okay, and the nucleolus is where these little yellow dots are created. Those little yellow dots are called ribosomes, and ribosomes are where proteins are assembled. So if we look back at your notes here, those are the last bits of notes. So the cell organelles, arguably the most distinct, outstanding one would be the nucleus. And it contains DNA, deoxyribonucleic acid, and it also contains the nucleus and the nucleolus, and that's where ribosomes are produced, and ribosomes are where proteins are made, and they're often attached to endoplasmic reticulum, which is a word which, if you can say it and spell it, you can win friends and influence people. Last bit about the nucleus, DNA. Yeah, it contains DNA. And if you take the DNA out of a cell, it's about 3 meters long. So you know those cells you looked at, microscopic, had tiny nuclei in them. Can you imagine putting 3 meters, one of those meter sticks, 3 of those long, curling that all up inside that nucleus? But that's what you're looking at. And we're going to talk a little bit more about that next time. I'm stopping at transcription and translation. I need to have enough time to go through this and show you some video clips of those. and they're going to be a little bit more involved. So make sure it'd be a good idea maybe to read those in the textbook before class on Monday, and we'll pick it up there on Monday. All right. Thank you.